that were just plots of 36 plants each with or without polymer, without any kind of close proximity to any of that would induce any kind of error. And from that, since we planted them at the beginning of last year, we did get preliminary results in the winter of 2009. What you can see is that already we have, well, the first consideration is that this is preliminary data with first-year plants. As Kai said, they don't grow into their max potential for 2 or 4 years. But already you can see that in the graphs of all the graphs, you can see that the plots from the fertile land and the plots from the marginal land are markedly different, showing that there is a difference between the growing capability on these two sites. But what you can also see is that in our plot of average weight, the polymer did have a marginal effect. And this is something we're going to be looking for in our future analysis. This is from a picture from our first year study. So even though this is our first year plant, it's still very juvenile. You can see that already it's growing to a very appreciable height, even within the first year. In addition to the field study, we also have a lab component. This is geared more to answering our second and third research questions. So in the lab, we have acquired a bomb calorimeter from the Belsa Agricultural Research Facility, which is under the FDA. And we're going to be performing what's known as bomb calorimetry on our miscanthus biomass sample. What this tells us is we essentially combust a sample and we measure how much energy it gives off in that combustion reaction. So it's a very easy, very quick, and very informative measure of exactly how much energy is readily available in the plant biomass in its raw form. We've also conducted soil analyses to quantify the differences in nutritional content between our two sites. We've also conducted statistical analyses in order to verify the differences, to verify that the findings that we found so far are significant, which they are. At the request of some of our experts that we've met with over the past year, we've added 2 additional components to our study. The first component is a pot study. Now, cross-linked polyacrylamide has been known to be able to absorb and remit water over time, but whether the plants are able to fully absorb the water out of the polymer, and whether the polymer's ability to absorb that water and release it over time is degraded, isn't well characterized. So this is a potential study that we're considering in the spring of next year that would help us quantify what kind of long-term effects of polymer rewetting that we can see and what the plant can effectively utilize it. The other study that we're planning is a root spread analysis, which is we would dig around our plot and see how far the roots have spread radially. The reason we're doing this is that even though miscanthus is a non-sterile, it's a sterile hybrid, the rhizomatic root system is still capable of moving outward, and if there's plants nearby or trees, this root system could choke or kill surrounding life. So it's something to be careful. With that, I'd like to hang it off to Ted Michael for our timeline and future plans.
Thanks, Mike. We're actually really excited about the next few semesters. As Kai mentioned, miscanthus doesn't reach its full potential for about 2 years. And so this upcoming harvest this winter will really allow us to do some much more telling analysis. As such, it's really going to be the bulk of the next semester, this fall and spring. And then in addition to that, this year's data will be much more informative from an economic standpoint, in terms of whether or not this is actually an economically viable or feasible option. We were also invited to present at the National Society of Black Engineers Region 2 Technical Research Conference, which went really well. Then we actually just got back from there. We also wanted to take some time to talk to you guys about sort of the Gemstone process, give you guys some advice about navigating the Gemstone program. As you can see, we're a relatively small team, with only 9 members. And as a result, we're really sort of a close-knit group. It's really important to always stay in touch with all of your team members. We're always on email, we're always on the phone, and this has actually made it a lot easier to handle the workload as everyone is sort of available to help each other with the different tasks as they go. With that said, I know some teams definitely like to, sort of separate into subgroups to get different things done. But, since we're sort of smaller, while we still distribute tasks sort of based on expertise, everyone is involved in everything, and everyone is always, reviewing each other's work. And so that helps to sort of keep everybody in the loop and make sure that, nothing gets overlooked. Everything we try and double check it, triple check it to make sure it's correct. Now I know you've all heard it before, and you'll hear it many, many times again. But start early and put as much into your literature review as possible. That will really make it so much easier to assemble the proposal and to assemble your thesis, and will allow you to be much more versatile when your project changes, and your project absolutely will change. In fact, one of our largest challenges was finding ways to implement some of the suggestions that our expert advisors had given. And having a really exhaustive background and literature review is really what made that possible. So with that, I just wanted to thank a couple people, and then we'll be happy to open it up for questions. Dr. Gary Felton, who's our mentor, obviously, Dr. John Leacox, who's helped us, Dr. Wild, the Beltsville Agricultural Research Lab, Urco, Irene Robinson, and obviously the Gemstone staff. So with that, thanks a lot, and we'll be happy to take some questions.